第一个问题我就问蒋博士，我就跟他说，我说，据我所知，这个驴子跟马，它的基因跟人差不多，它没有那个基因会基因之基因会合成这个翻译中文好了，因为等会儿都是英文啊，这个就是纤维素酶。你有纤维素酶的话，你才能够消化这个稻草，吃这个稻草。哎，人没有这个，他我说这个是不是寄生来的？这是共生来的。他说是的，第一。然后我第二个问题问他，我说在每次饥荒的时候，很多人这个很苦的说要吃这个树皮，当时大部分人都死掉，是不是有小部分人5%、5%、20%都活下来？他是不是有不同的情形？对，这个他立刻就同意。那时候我就决定说要请他来，为什么？第一，他是我们这个 ABC 的第2代，我们这个会都是平均年龄，但是大概快要65岁，对不起，我刚刚讲实际年龄，对不对？然后呢，我们要需要这个生力军。当然，我们这个他带来的这个朋友他昨天给我写的名字，他叫黄美仙。那黄美仙他说他是75%的不是Chinese，25%的 Chinese。但是我觉得他的 knowledge75%是 Chinese， 我说这不是，所以他也是一个 pharmacist， 我也欢迎他参加我们的会，这是这个要求的。等会儿他要欢迎他上来的时候来讲的话，这个就大家拍手，这个荣耀，王兆标在他去年会的时候，他讲这个演讲要用英文，那个演讲要用英文，之后他结果通通落空。所以我现在今天找到贾立平，主要是说他根本不会讲中文，所以他绝对不会落空，对不对？然后我就把名字给他了，这个我就给了，这不是 Big House 讲，给了他，王兆标又来了，他中文名字怎么讲？我说要中文名字干什么东西呢？然后中文名字我就抓瞎子了，我就根本不知道，我大概猜到他甲，猜到他平，中间那个字我不知道，那个立这个中国字很多，厉害的厉，什么的厉，名利的利，对不对？然后我就问他，哎，这个他忙不得了，我看他的机会不多，虽然我们不是很远，大概不到1里路，但是他说，哎，他会写6，1、2、3、4、5、6。他说6的那个2个那个腿是这样子的，我立刻就知道，我就合过来变成马上就16给他，他立刻就说对，给他厉害的地震的地啊，对，我给大家介绍的时候不用什么像。academic 介绍他哪里毕业，发表多少文章，那个谁都没有人要听，大家比较喜欢一点，对不对？对了，这个至于这个时候，那时候这个团长立刻就说，这种吸取一定对人有这个有帮助，有什么好处？我说慢点，我们科学只是用这个基因测验它中间这个跟人不一样，但是它跟这个人千万年出来，当时没有看也有做一点好事，我也刚刚跟这个贾博士请教过，他说没有，很多地方这个病人怎么样治不好，然后跑到没有坑里一下才能好，怎么样？他把别人大便弄到他的肠子里，接触到他肠子里。Sorry, if you don't understand Chinese, I personal translate for you later on. 然后所以这个是非常有意思的事情。所以我们先等会儿介绍，我就不要占他的太多的时间，让他来讲。他开始的时候，我们就给了大家好好的鼓掌，鼓励他，也鼓励就是这个介绍他们写的名字很好，说王美仙，这个美丽的美，仙人的仙，他是个 pharmacist。欢迎他们参加我们这个会，给大家。
你好，我是贾立平。 program at Mayo Clinic. And what the microbiome program studies is how the microbes in your body, all over your body, how they work in concert with your body and how you live in ecological harmony with them to promote health, and how when they're in imbalance, they can promote disease. And this is actually a topic that's received a lot of attention in Chinese medicine and in China, with huge efforts from the Beijing Genome Institute and other places all over Asia, Singapore and so on. And this is a very attractive principle. I think we have to be very careful about it because there's a very strong Chinese bias towards this idea of living in harmony with your environment. But there's a lot of promise here as well. So I think there's a lot of cautious optimism on both fronts. And what we're doing in Mayo Clinic isn't on the, the focus isn't Chinese medicine, but it's this ecological principle that we're chasing down. So I'm part of the Center for Individualized Medicine. There are 5 branches. You will notice that all of them involve genomes in some way, lots of data, and that's my specialty. I started as a physicist. I did my training in statistics of sequence alignment before anyone knew what biophysics was. My thesis committee passed me because they had no idea what to do with me. Then I went on to study microbiology as a physicist and ended up in Mayo Clinic studying the microbiome. And you'll notice that if you look at these 5 programs, clinomics, epigenomics, which is sequencing the methylation pattern in your genome, biomarker discovery, which is sequencing the human genome and looking for markers in the human genome of disease, clinomics, which is chasing through the clinical data and trying to understand correlations with your gene sequences, and pharmacogenomics is the same thing, looking for biomarkers of certain genes that will tell you how you react to certain drugs. The microbiome is the only one that doesn't involve the human genome. So we are off on the side. We're kind of this interesting program. But why in this program that's so focused on individualized medicine, where the human genome is key, is the microbiome so important? So I'm going to tell you why you should care about your microbiome. So first of all, it's a microbial world. You think perhaps everything you see dominates the world, the trees, us, the grass. What you really see is that the number of people on Earth is something in the billions, right? 10 to the 9. So that's about 7 billion people. The microbes in your GI tract have about 13 more zeros behind them. And in fact, if you do a count of the number of human cells you have in your body versus the number of microbial cells you have in your body, you have about 10 times as many microbes as you do human cells. So there's this little joke we have about who's really, serving who here. We might just be vehicles for our microbiomes. And then if you compare this with number of stars in the galaxy, or known stars in the universe, it's 10 to the 24, which is 2 more zeros than just your GI tract. And if you compare that to the number of microbes we think there are in the world, we just count based on ocean, soil, and just take a quick estimate, that's 10 to the 30. So that outstrips by a factor of 1,000,000 the number of stars in the universe. So they make up the majority of diversity, they make up the majority of life, they make up the majority of metabolism, and they make up most of the important interactions that occur on the planet, but we don't see them, so we don't think about them very often. But nonetheless, it's a microbial world, and if you think that it just applies to the environment, you'd be wrong. So there are a lot of different genes, as we said. If you look, there are 10 times as, or 1,000 times as many different types of genes. There obviously are a lot more genomes in our GI tract, about 100 different species at least. Counts vary between 100 and 1,000. The number of metabolic reactions. So we have certain metabolisms built into us, but we rely on our microbes to produce certain vitamins, certain cofactors. We rely on microbes in our environment to do anything from break down waste, decompose things. But the variety of metabolisms and functions they assess is huge. Microbes have been known to eat nuclear waste. They have been known to eat antibiotics. They can do almost anything you can think of, they can turn into a food source. They're really pretty amazing. So, before you think it's only an environmental problem and they only dominate the environment, they actually dominate us as well. So if you look at this chart, these are the leading causes of death according to the WHO in 2004. And a number of them are directly infection-based, right? So the infections are highlighted in yellow, and that's not even the microbiome. That's just considered infectious disease. But if you look at many of the other diseases, they've been linked to the microbiome. So lung cancers, prematurity, and low birth weight, COPD, stroke, coronary heart disease, all of these have been linked to the microbiome through the microbes' interaction and ability to digest nutrients, or to form calcifications, or to influence the lung environment and basically degrade the lining of your lung. So let's see, about now, I guess 8 years ago now, the National Institute of Health put out a $150 million 5-year program called the Human Microbiome Program. And that program was meant to explore the unknown territory of the human microbiome. We did not have any information. So this is almost purely unscientific. It was just survey. We had no idea what was there, but we knew it was there. And we decided we had to find out what it was. So we went by site and tried to find out what all the microbes were by site. We went through 7 different sites, and including the eye, which didn't really have that much of a microbiome. But they went through blood, through the urogenital tract, through the airways, the skin, the oral, and the GI tract. And the GI tract turns out to be one of the main focuses and one of the most stable microbiomes there is, and now is probably the predominant focus of research. The Institute of Medicine in 2012 announced this as integral to human health and physiology, that we need to understand this if we're going to understand what makes up a healthy human being. So this has received a lot of attention. You've probably seen this, I think between 2012 and 2013, the microbiome came on almost the cover of every single magazine, including The Economist, and is on the New York Times and all the science magazines, of course. So this has gotten a lot of attention, and in a number of diseases. Obesity has been huge, obesity and malnutrition. So, the microbes get to your food first, before you do. You just get what's left. Neurological diseases, autism, and so on. Any autoimmune disease or impaired immune response. It's gotten a lot of attention in multiple sclerosis, rheumatoid arthritis, and gastric cancers. We know the H. pylori story, where H. pylori has been found to be causative of stomach cancer, and my particular research program is focused on colorectal cancer and the link with the microbiome program. So to summarize, you have 100 trillion microbes in your gut. They convert the indigestible food components. They do things like stimulate your immune system, maintain your health. There's a link between any number of host factors, both signaling and metabolic, and they've been implicated in a lot of non-infectious diseases. So there is a very nice voice here, but we don't have sound, so I'm going to try to do the best I can. And this is to introduce the microbiome. This is a little video we put together with a voice that sounds like it's from the Discovery Channel. You just got me. But anyway, the microbiome is in several different environments, and all over your airway, mouth, skin, and we're learning more and more about how this impacts your health. And this is going to display an example in the GI tract, where if you go into the GI tract, we have multiple species, and they live in balance. This is the ecological harmony of the microbiome. And when you have a GI problem, the common approach to this is to say, well, there's a problem pathogen, so let me just take antibiotics and wipe them out. Now, so you think, oh, I have an infection, let me just wipe them out with antibiotics. Now the problem with that is we know now that the ecological balance was so important that if you knock things out with antibiotics, you might disturb the microbiome in a way that leads to a different problem. And in this case, the example is C. difficile infection. Because your microbiome, your healthy microbiome, is preventing other infections from coming in. So this is a severe problem. In fact, antibiotic treatment has been linked to C. diff now, and now we know that by restoring the microbiome, by introducing somebody else's microbiome to yours, we can restore health. And this is an approach that Mayo Clinic and other places have used with about a 95% success rate, where we just take somebody else's GI tract and introduce it into yours. And now we're coming up with drugs for that are probiotics. Now my part is actually the genomics, so this is just a highlight of the genomics and the fact that we're using genomic data to figure this out. But I'll talk more about that as we go along. And these are pictures of metabolic pathways. So just to quickly summarize, what did we learn? So the gut microbiome provides vital functions. It converts indigestible foods, such as oligosaccharides into sugars that you can use. Produces essential cofactors and vitamins that otherwise you can't produce. Right? You require your microbiome, in fact. And if you don't have a microbiome, you don't develop a proper immune system. So, and it's believed that if you don't have a proper microbiome, you may end up with several autoimmune diseases. And in addition to that, your microbiome metabolizes medicines. Imbalances lead to diseases such as C. diff, obesity, colon cancer, and GI dysmotility. So what's the goal of the research program? The goal of our research program is to take unhealthy microbiomes, which this is a rough sketch. This is often a kind of plot you'll see where we can put the points for each microbiome community into space in relation to each other. And we can see, for example, that healthy might represent a certain state, and they might all hover around a certain space, and diseases will be in different places. And the goal is to understand, how do we restore health? What is health? How do we restore it? Do we get there through transplantation of the microbiome? Do we get there through antibiotics, prebiotics, which are foods, and probiotics, which are microbes that we grow and give you in pill form? Or you may have seen them in the supermarket. So how do we do it? So I'm going to describe our roadmap to the clinic. There are several steps in this roadmap. The first step is coming up with a pilot and a hypothesis. Then we collect data, then we validate the data, test in models, and treatment. So the first step on the pilot, we come up with an idea, or we come up with a survey that we want to do. Sometimes the survey has a hypothesis behind it, sometimes it doesn't. So an example might be, I think the example I've listed here is rheumatoid arthritis. And the thought is that, well, we've seen links in pilot data between rheumatoid arthritis and the microbiome, so we know that there may be some kind of causal effect. So we want to test this. So, and it's not just rheumatoid arthritis. We actually serve a number of different programs. So here are some of the diseases that we're working on, and the level of pilot funding or publication or extramural funding that they've received. So it's pretty diverse. And here's our pilot processing pipeline. So this is a pipeline that I've put together, both to collect, to extract the DNA, to sequence the DNA, and then to process that information through a process that can assist the clinicians with their publication and statistics. And this involves a team of, I'd say about 20 people. But once everything is linked up, it becomes a machine that churns through pilot data really quickly to see if there's something interesting there. Then if we have a hypothesis that seems to work, say we do 20 and 20, we take 40 people or we take 50 people and we see something interesting, then we'd say, well, let's look at that in more detail. Let's try to find out what's going on. And that's where we can really employ the Mayo Clinic Biobank and Serum database. You know, Mayo has these huge infrastructure resources, and we're turning them around to the microbiome program, where now we're collecting a lot of stool samples, which are proxy to the GI tract, in addition to blood and tissue. And we've incorporated into this biobank, which has a huge number of subjects enrolled. So the biobank's a facility that's about 35,000 square feet. It is essentially an entire floor plan. It has automated machines that basically can freeze and take tissue and you know, robotically manage a freezer. So it will both freeze the sample, store the sample, and retrieve the sample, all by just dialing it in. And there's a new space out in the warehouse out in 41st Street for this. It involves all 3 sites, and it's heavily linked with pathology and with the biospecimen accessioning and processing core. So these are things that, from a research perspective, we all share and we can all dial in. So just to give you an idea of the volumes, and these are important when you're validating your hypotheses, we've collected, I guess, about 57,000 from 57,000 patients or so. We've aliquoted a number of different samples, whether they're blood or they're tissue or whatever. And probably in the microbiome world, we probably have about 10,000 samples or so relevant to that. It's because we're just starting, but this is picking up really quickly. To go along with this, we have a lot of tissue sections, right? So we have paraffin embedded, and we have the ability to use laser capture microdissection to pick up certain parts. And this becomes an interesting challenge in the microbiome area because, of course, the GI tract is made up of many different places, and you want to understand the interaction, for me at least, depending on what your study is, but many of us want to understand the interaction at the epithelial layer, right? The layer between the microbe and the cell, and the human cells. So this is the robot that you saw before that I described. This is really what's helped us quite a bit, and we've been expanding and getting a number of these freezers, thanks to Steve Thibodeau and the Biobank service. So the goal is to enroll 50,000 Mayo patients, regardless of medical history, so that any study you would want to do, you would be able to say, for example, for me, I would be interested in colorectal cancer. I'd be able to say, could you tell me who has colorectal cancer in your cohort that I could go back to and collect data on? You've already collected their sample, now can I use it for science? And likewise, I can say, well, I need gender and age matched controls with similar diets. I can do that. And I can do that as soon as the hypothesis is formed, right? I don't have to do a collection each time because these are collected and stored already. So these are some interesting details for study purposes, but they're consent forms. They allow us to go back to the data. If you join the Mayo Biobank, if any of you have joined, you get a packet this thick. It's got a large consent form. It's got a lot of questions in it. And that really helps us a lot, right? I've looked at it as well. It's a long form. It takes a long time to fill out, but it's really very helpful from our perspective to be able to go through and mine through the data. And it comes in several forms. We try to make it as easy as possible. We provide paper, it's scannable, it can be done on the web, and if you really, really have trouble, I think you can even call them on the phone, and they will help you fill it out. Okay, so blood is another area where the biobank has extensive collections, and that is quite important for a number of reasons. One is they collect they break the blood into different fragments, and some of these fragments are very good for understanding how the immune system works, and the immune system is one of the main interactions we believe you have with your microbiome. It's the main way you regulate your microbiome, and it's the main way your microbiome regulates you. So you never know which way it goes. Some of the examples of regulation, just as an aside, people believe that your temperature, your core temperature, may be regulated by your microbiome. Right? Without your microbiome, you would be a few degrees cooler. So it's an interesting interplay. So a lot of the strengths here, this is embedded in the clinical practice. This happens day to day. Mayo has a high volume of patients every year, and we pick up some of these for research, and really helps us speed things along. We get to leverage, as such, we get to leverage the electronic medical record, and we're building natural language processing software to be able to mine that. Because of course your doctor writes down their own notes, and they take down their own dictation. And that means that not everything is a checkbox, right? In many ways, the doctor's already performing individualized medicine. It's already tailored to you, and so are their reports. That means that we actually have to go through and come up with natural language processing so we can read their reports and understand what they mean when we're using them. We can reuse data, and we can interface with Rochester epidemiology, which already has a very large program and a very successful Olmsted County cohort. So the next step after that is to validate the data. So we want to confirm the hypotheses and analyze the data sets. And I'm just going to talk a little bit about some of the tools we use for that. So one of the tools is a 16S rRNA marker. rRNA stands for ribosomal RNA, and 16S is a particular subunit of the ribosomal complex inside of microbes. And every single microbe has one. And they all look more or less like this, but they all have slightly different sequences. And we can use this molecule to trace back about 4 billion years in evolution. So we can get the relations of the different microbes. And what we can do then is understand who is in your microbiome with this really simple test. And the way we do that is we sequence certain regions of this molecule. We pick a certain region, in this case, let's say V8. Normally we do V3 to V5, but this illustration has V8. And we sequence that region for everything in your microbiome. And we get different sequences given by these different colors, right? And if we count up the different reads, we'll say, well, you know, there are 10 of the blue ones, there are 1,000 of the sort of reddish-orange ones, and there are 800 of the magenta ones. And that gives us an idea of not only who is there, but how much they're there. What's their relative abundance? So I know that, you know, the reddish-orange one is 100 times more present than the light blue. And therefore I know how many microbes you have. I know you have 1,000 bacteroides versus, you know, 10 Clostridia or something like that. And that can give me a baseline idea of what's going on. And the reason why this becomes possible is because we have next generation sequencing. Where people used to have molecular assays and a lot of work, now we just hit it with a hammer. And then we worry about the computation on the back end. Right? So we can do that because the costs have gone down. It's, for base pairs, it's fractions of a penny per base pair at this point. And we have 2 lines of technology, both the Illumina and the 454. The 454 is getting outdated, but if you look at the cost, 454 was produced 450 base pair reads and about 500,000 of them, and that would cost you about 10 or 20,000. Now with Illumina, it now produces, actually this is a little bit outdated, it now produces about 600 million reads at about 150 million base pairs, 150 base pairs per read. So the cost just keeps dropping. In fact, we don't even try to load the sequencer as much as we can anymore because we would just be waiting too long. And this costs about 2,000, I should say, 2,500 a run. So we're really dropping the cost down. Now, what do we do with this information? So I'm going to show a couple of the cute little tools we use. One of them is to compare different communities. We actually use a little tool from computer science. I don't know how many of you might be familiar with computer science and compression algorithms, but whenever you zip up a file, what the computer is doing is it's trying to say, let me look for the repetitive structure inside of your file. Like, you just keep writing the same thing over and over again. The computer doesn't care. It uses that to compress your file. So instead of saying, well, gee, this has repetitive structure, it says I can use that and make the file 10 times smaller. And we can do the same thing. And we can put two files together and ask the computer to compress them. And if the two files are very similar to each other, they'll end up compressing very well. In other words, those two microbiome communities are very similar to each other. You'll get a very good compression ratio. Now, if those two communities are completely different, like I'm looking at soil and then I'm looking at ocean, those two communities are not going to overlap very much. And the information between those is not going to be compressible. And we can use that as a proxy for distance between communities. The other incredibly long acronym, which I'm not going to go over, is IM-Tornado, which is a tool for processing these reads and spitting out several bits of information. And the main thing this tool is it does a lot of bookkeeping, because what Illumina actually does is it produces two fragments, one on each side of the DNA, and there's a gap in the middle quite often. And when there's a gap in the middle, more or less people don't know what to do with it, because it's much easier to process things if there's just one read, right? But if you have a gap, then you have this conundrum of how do you process all your information and keep track of what's going on. You certainly could process them both and do everything, and then keep the links. But because of how large these data sets are, you really want to speed up your calculations, and so you quite often use tricks like compressing one side and compressing the other side and then running them separately, but then you have to join them all up. And this basically runs through these calculations but keeps track of what's going on each side, and so you can bring them back together. And it produces both a list of taxonomy, what we call clustering, which is an organizational taxonomic unit, and a phylogenetic tree, which are all important for subsequent discovery. So that's some of our validation process. Next, we get into the modeling, and the modeling has two real forms. When we say we want to go to a model, we don't just want to understand who's there and what the link is. Now we want to understand why it's happening. Because if we understand why it's happening, that tells us something about the disease. The microbiome side and validation can be good for blind treatments, which sometimes work, right? When we say, well, we don't know why this treatment would work, but we know that this microbe is correlated with health, so we'll use it. But ultimately, we want to understand why that is, and we try to test these things in models. And the models come in two forms. They generally come in mouse form, or they come in computer form. I specialize in the computer form. I'll tell you a little bit about the mouse form, just because we do have some of that in our program. So this is an example. This is actually my plot. This is an example where we fed mice both normal food and high-fat food. So normal diet and a high-fat diet. And then we said, well, we might exercise them or not exercise them. So they put these mice on these treadmills. And it was a forced exercise, because the mouse was either running or it was spinning. And the other mice, we let them sit there and do nothing. So, and what you see in this experiment is that there's a big separator from diet, and there's a separate separator from exercise. So both exercise and diet affect your microbiome differently. And what was interesting about this was these have a known effect on your cognition and your behavior. So the mice that were on normal diet and exercise are less anxious and they have higher cognition. And the mice that are fed a high-fat diet have sort of low cognitive ability and are highly anxious. There are other experiments we do in the Notobiotic facility. Now these are very interesting experiments because what these are used for is to humanize mice. What we do is we take these mice and we put them in bubbles. And they're bred so that there are no microbes in their environment for their entire life. So they have no microbes, and we get the chance to introduce microbes to them as we wish. So if we think a microbe has a certain effect, we can choose which microbes we will give to this mouse. And quite often we use this for humanized mice, where instead of them having a native mouse microbiome, we will introduce them a human microbiome. And then we can add things to it, subtract things to it, and have fuller control. And actually, these germ-free mice are where a lot of our conclusions come from. We know that when we raise germ-free mice from a young age with no microbiome, they have a compromised immune system. It just never develops. They have a lower temperature. They never digest their foods really well. They have low weight. They have all these issues. Okay, but now I'm going to talk about the computational modeling, which is really my side of things. And I'm just going to give you a quick example. And feel free to stop me with any questions. So we often want to talk about metabolism, right? So say we want to know what's going on, we want to talk about what's being digested. What is the food doing? And so we'll say, well, why don't we use metabolic modeling, right? An aspect of systems biology to try to measure this. And what a metabolic model is it's an entire reconstruction. I don't think you can see it very well, but it's a large map. Typical microbe contains about 2,000, 3,000 reactions, about 2,000, 3,000 metabolites, and they're all drawn together in a network from which you can understand the flow. And you can understand what reaction is creating what down the chain of the entire metabolic chain. And you can understand what is leading to reproduction and what byproducts that microbe is excreting and what food it's uptaking. Now, we often have issues in reconstructions. We often start with a parts list, and there's some missing reactions, right? You start listing the parts, you start attaching the reactions to each other, and there are a bunch of missing ones. And the challenge there is to figure out what's missing. So here's an example. You know that you want to get, let's say, from, this is in Illinois, say you want to get from the Illinois Union to Cranor, right, which is a concert hall, because you're a professor who just can't miss concerts every week, and you want to know how to get there, and each street has a certain directionality, and what's the best path? So in green is the best path, right? And there are a couple of different paths you could take, but that one's the best one. And that's what we want to be able to tell you out of a metabolic network. So, but there are a lot of things that can go wrong. So inside of every element, every step we have, there are mistakes that can be made, and these are things that we need to be able to account for. So you get your DNA reads, you try to put them together into a genome. Well, there are a lot of assembly errors you can make through that. So what you're doing is you're trying to decide if two reads are adjacent to each other, but it might belong somewhere else as well. So it's like taking pages out of a book. Let's say you have 10 books, and I tore all the pages out and I put them on the floor. I said, now put the book back in order. Well, you might think that, hey, this sentence joins up with this one over here, and most of the time you might get it right. But sometimes you would mix it up, and you would have the wrong books, especially if the books were similar to each other. So we have the same problem. We can have badly called genes, where we just mistakenly say there's a reaction in the genome that doesn't belong there, or we miss something, right? That can happen. Or incorrect ambiguous functions. That's another thing that can happen through the genes. And we can therefore miss these biochemical links. So then when we go through our network, all of a sudden it makes no sense, because we're going from point A to point B. We know point B happens, but there's no road in between those. So that's exactly what happens, is we have, a working model would look like this, but a broken model would look, like this side, because there's no way of getting to Cranor. There's just a one-way road that goes the wrong way. So when you have that, what are you supposed to do? Well, what we do is we actually go through and we fix the model, right? And we do that through what's called gap filling. And what gap filling does is it tries to identify what's the most likely missing link. Based on other information from other genomes that I have and other reactions that are out in the database, we know that certain reactions exist in nature. And if it exists in nature and it solves your problem, it may be in this model. Now, how do people usually do this? Well, the old-fashioned way is manual curation, right? This is the first way. Paulson's lab did it. Paulson is basically the grandfather of metabolic modeling. He had graduate students and undergraduate students, and they sat down for the first E. coli model for 2 years and read every single paper on E. coli biochemistry and wrote it all down. Now, we'd like not to do that because it kind of sucks. And you'll never get anywhere if you have to do that with every single organism, because your microbiome has about 100 species in it. So we have to come up with a better way of doing this, and it doesn't scale, right? So now estimates, even with the knowledge we have and with the expertise we have, the current estimate for a single microbe is about 6 months. It would take you about 6 months to put together a model by hand. So we have computational gap filling. And that was an algorithm first written by Costas Moranis. And basically what he did was he did a parsimony approach. He said, let me take the shortest path to close a gap. That should work, right? And it kind of does work, but it has a couple of issues. So one of the issues is that biology does not have to obey parsimony, right? We are not built around efficiency. Biology evolves, which means it always has to work, and that it's not necessarily a well-designed system. This is not a computer chip we're designing, right? This is an ad hoc system. This is like your house. You don't raise your house and redesign it every time you want to incorporate a room. You add a sunroom, it probably doesn't have insulation under the basement. Its electrical system is, you know, some add-on to your old box, and that's how the cell has worked for billions of years. It has been adding on and subtracting and adding on and replacing things in not so neat modules. So it's like an old house that's been modified many times. And parsimony is assuming that it's just been built for efficiency, and that's wrong, right? And so what can happen is you really have 2 or 3 reactions in the place of 1, because maybe it worked better that way. Maybe it was easier to pipe, you know, the water through, you know, some adjacent bathroom instead of piping it through the main. And that's the ultimate problem we have. But we have a trace of this evolutionarily through the organism's genome and the relatedness with the other genes. And we can use that to try to determine how likely each reaction is. And by doing that and using that information, we can say, you know, this top path, you know, there's really only a 5% chance that these genes are involved, but in the bottom path, we really see much more likelihood, and that reaction set seems to make more sense. So it's a better way of estimating reactions. And the way we get those evidence scores is by looking at the homology of the genes in the database. So what we do is we say, well, let's look for how much evidence there is. So red is high evidence, blue is low evidence. And there are a couple of different mistakes you can have. You can have evidence supporting A, but a little bit of evidence supporting B. And that would get ignored by many of your annotation engines, because it would say, well, A is the function that makes the most sense, so that's it. Whereas a probability function would tell you, well, it could be A, mostly A, but there's a little chance it'll be B. So B makes more sense in your network, let's do that. It also can be evidence below the threshold. So an annotation engine will often, let's say A is very weak evidence for it, it will just miss that gene. Because it says, I don't have enough evidence to call it, but between the homology evidence and your network, if it makes sense, then you should put it in. And there can be conflicting evidence. There are a number of genes that are very similar to each other, but have different functions, right? They might transport magnesium instead of chlorine, right? Or calcium. And it's very easy for a gene that does transport to change the ion it transports. But that can be very different for the cell. And this takes into account that as well. So this is our scoring function. Basically, this is the methodology. We have a gap filling formulation. Basically, it says optimize the likelihood of the reactions inside of the reaction network while reaching growth. The cell has to grow or reach its objective function, whatever we define. And microbes have this nice feature that they're always trying to grow. They like to replicate. So that helps us out a lot. And then there are a couple of other thermodynamic penalties, database-related penalties, where we say this database is worse than the other one, so we trust this one more, and things like that. And at the end of the day, we can get a curve, and what this, so this is a response curve. And when we order the reactions, the way to read this is that the ideal curve would go straight up and then over. So the more we're like straight up and over, the better we're doing. And what that would mean, if it was straight up, would mean all of the reactions that are right would come first, And every time we go horizontal, that's a reaction that we get wrong. And the order they happen in is very important. So what this is showing is that for two very key models that we know the right answer for, or most of the right answer, we do quite often get the right answer before we get a wrong answer. So all the right answers have the higher likelihoods. So, the summary is that finding the alternates is valuable, and the true positives were not always the most likely annotations. So what that's saying is that quite often, even if you have a very high homology, that might not be the best reaction to have based on your network structure. So tools like this are very important. And this is more likely to be useful in organisms that have some relation to something in the database. Okay, and so we've implemented this in this large knowledge base. So knowledge base is actually this other large multi-million-dollar project. The Department of Energy sponsored this large $100 million project for systems biology called knowledge base. And what knowledge base is the collection of all databases put into a uniform, usable, API-frozen service so that people could build their algorithms on top. And the Department of Energy had the foresight to do this because they said, you know, there's so many research groups we sponsor that have to face the same challenges, that have to come up with their own databases, that have to write their own tools, that have to maintain their own infrastructure. Why don't we just pay the government labs to just, you know, why don't we just have the government labs create this large service? So we actually brought this up to them and we said, well, you know, we have this idea and we think it will work really well. And they actually were very nice and implemented our tool inside of knowledge base. We presented it to them, went to them, and they said, you know what, your tool is going to be the next generation tool, so why don't we just use it and use it as a replacement for the metabolic services that currently exist inside of knowledge base. So this has been quite good for us because it's made our tool very accessible and to the entire community and has been a big boost. It basically makes it very easy to test multiple hypotheses. If you have a metabolic network, you can easily test whether a certain metabolism is happening, whether a certain compound is getting digested or produced. And the long-term vision of this, just to trace through, is that we're going to start with the 16S rRNA survey. And what that's going to enable us to do is understand who's there. And from there, we're going to, if we come up with a hypothesis, go after the microbial genomes and assemble those and be able to say what they're doing. Right? What functions are they carrying out by metabolically modeling them? And then ultimately, through the metabolic modeling, which is what I just presented to you, we're going to come to an understanding of the community level. So in other words, now that we can understand each individual organism, let's put them together as a community and understand what the entire GI tract is doing and how that's interfacing with your body to promote health and disease. And just as a quick advertisement, you know, since we've replaced their service, we now have about almost 20,000 models that are posted on KBase that are publicly available, that have essentially become the global standard for metabolic model repository. So that's very exciting. So finally, I just want to put a brief note on how we move from mechanism over into the clinic, and how do we move service lines down. And this involves a lot of regulation and CLIA certification and what is otherwise very boring stuff, but very key to science. And we're actually finding some of the more basic scientific questions. So, you know, often when you're in a university, you don't, I only appreciate this now I'm in Mayo. So often when you're in a university, you don't really think about how to reproduce something as much as you think about it when you get to a clinical practice. You know, science is supposed to be about reproducibility, but ultimately, are you going to do the same experiment 3 times, or are you going to move on to a new experiment? Well, when you have a clinical standard, you have to be able to reproduce the results over and over again, which means every aspect of your assay has to be locked down. So we have studies in all of these areas now. In other words, we have studies that are trying to understand what's the effect of different collection methodologies, right? And what's the effect of let's say we send out a home collection kit and we tell you, hey, put this in the freezer, and you decide not to put it in the freezer for some reason, right? I mean, how can we detect that? Or what's the effect of it? Does it make a difference? Should we be giving you some kind of solution or buffer that makes it so that it doesn't really make a difference, even if you do make a mistake? You know, we have storage, right? How should we store it? Should it be frozen? Should it not be frozen? What happens if it sits on the dock for a while? Right? These are all things we have to worry about once we have a clinical test, because these can happen to real people. And then there's more mundane laboratory things, DNA extraction, right? Steps like that and steps like amplification. Right? How does that change over time? How do we make sure it hasn't changed? How do we create a standard, like a sort of a gold standard microbiome that we can use as an internal control to make sure that nothing has gone wrong with the sequencing, with the informatics technology, or the analysis? Now the computer side is actually much easier because we can control that pretty well. But all of these need to be understood before we can move forward. And the main challenge with the computational side is actually the time, right? Because we have to return these results within a certain time frame, and the computations through millions of reads can be quite intensive. Quick summary, our microbial partners are very important. They're a big part of our community. It's imperative we understand their role in health and disease, and what we're doing now is we're creating a pathway to the clinic that gets us through all the steps we need to get to treatment. Now, what are we doing with it? So I want to give you a quick example, and I think I've got just a little bit of time. But so a quick example is my particular program focuses on colorectal cancer. Now, colorectal cancer is a very interesting case because it's about 15% genetic. I would say people say between 5 and 20% genetic at most, right? Which means it's about 85% environmental. Now, how do you interface with your environment? The microbes are not separable from your environment. They are your interface with the environment. They get first crack at the metabolites. They are the major players in signaling. They produce everything that interacts with your body, right? And compounds interact with your body in a way, but the microbiome, by and large, is part of that environment and very much the mediator of that environment. So when we say something is environmental, especially in the GI tract, we're really saying it's microbiome. And we know that this is the case to some degree because we have evidence for it. We have the effect of diet on colorectal cancer rate. We know that colorectal cancer in Africa is extremely rare. It's not a problem. We believe that the diet, but they have a very different diet, right? Their diet is not as much meat-based, it's more fiber-based. Whereas the African-American population in the US, which is on a Western diet, has the highest incidence rate out of all racial groups inside of the United States. Well, we've taken a baseline, gene set, and we've moved it from one place to another, which has changed its environment, and we've given it McDonald's, right? We have Alaskan natives, which around the world have the highest incidence rate of colorectal cancer. It's something like 10%. I was trying to look it up, but it's ridiculous. It's ridiculously high. If you look at polyps, like 50% of the population have these deep polyps. And what do they eat? They eat whale fat and more whale fat, I think. Mostly like seal fat, whale fat, fish. I mean, their diet is very monotonous, and it's highly fat-based, and with fish as well. And we know that the microbiome digests food, but it also produces toxins that may be the cause of cancer, and that's what I focus on. So my main focus is in this little part over here, and I'm interested in how the bacteria digest certain products and produce things such as hydrogen sulfide or other toxins. And hydrogen sulfide is an interesting example because it's known to be a genotoxic agent, right? And actually our bodies produce it endogenously as well, but so can the microbiome. And we know that when we take hydrogen sulfide and we introduce it to DNA, it mutates it. And we believe that through this and through other mechanisms that also can produce damage to your epithelial layer, that you accumulate damage, perhaps if you have the wrong microbiome. And kind of like UV radiation would cause damage to your skin over time, your microbiome may be producing products that damage your epithelial lining over time and causing cancer. So with that, I'm just going to wrap up real quick. You know, we need to understand what's going on so we can make an impact on the patient and establish biomarkers and design therapeutics. And just close out with some acknowledgments to my program. Our director, Jean-Rico Ferrugia, and my director, Heidi Nelson, along with our collaborators and partners. And this is my lab, and those are the Mayo brothers. And my funding agencies. Thank you very much.
Yeah, I think my question is, you have done all these wonderful research. I'm just curious, how does a doctor apply all these knowledge to help the patient? For example, if someone comes in to Mayo Clinic to see a doctor who thinks he has some problem, but after all these, tests, the doctor couldn't determine what the problem is. So is it by that time the doctor will be thinking, maybe I will take someone else's stuff and put in this guy's stuff? Maybe that will work.
No, that's a good question. And so I sort of glossed around it, but because we just ate. But we do fecal microbiome transplants.
Oh, really?
Where what we do is we take stool from one person and, introduce it into another person. And that works really well in the case of C. diff. So it knocks out that infection with a 95% success rate. And these are patients that have been suffering for a long time. They can't get rid of the infection. It's resistant, and it just keeps reoccurring. And the microbiome transplant ends up working quite well. There are other problems where clinicians do turn to those or to probiotics. I would say that at Mayo Clinic, it's a little bit more conservative, so there are other institutes where they will say, oh, what the hey, let's just try it, right? Child will be suffering from autism, they'll say, oh, what the hey, let's just try it. At Mayo Clinic, we'd probably do a trial first. And then, I'm just saying this because, and I think different countries have different stances. I swear there's this one doctor in Australia that just gives fecal microbiome transplants for anything, and writes a paper on it. I mean, you could be suffering from like a cavity or something, and he would give you a fecal microbiome transplant. But there are ultimately different tactics, and I think at Mayo we're a little bit more conservative. There are some groups that will try probiotics. There are some known probiotics. By and large, we won't just do it. But there are a lot of places around the world where they will do this this kind of treatment for anything from Parkinson's to multiple sclerosis to autism to other diseases. IBD is a big one that Mayo's thinking about but hasn't acted on yet. So it takes us a while before we get there. And our goal is, our belief is that because the risk of taking someone else's microbiome is high, we really don't want to implement it without a very severe reason or without control. So we're working with drug companies to produce known mixes of microbes that we grow in the laboratory, so we know exactly what we're giving to the patient before we treat them.
For me to think
just
about,
one quick question.
just a second. For me, think about the very simple thing. How so many things. I heard one thing is true. 黄金万两, 黄金, it's not 万两. Mayo Clinic is not going to put in the bank. Right? That's true. So go ahead for your question.
Okay, for your profession.
Don't too deep.
Very simple. From what you studied and you researched on, is it important or even helpful for one to obtain their parents' medical information, or for us to pass our information down to the next generation? Is that helpful?
So it's very helpful from the viewpoint of almost any disease, or any clinician would want to know that information. From the microbiome aspect, we don't know fully, but we do believe that it is passed on, especially if you're living in the same household. we only have ancillary bits of evidence. So the only study I can point you to is actually one with households with dogs and without dogs. So apparently if you have a dog, you and your spouse share more of your microbiome than if you don't have a dog. So apparently this dog is healthy. I mean, we know that it matters. We don't know how much, but this is the nature of the beast right now. We just don't have every variable.
So you mentioned about mice that doesn't have in a controlled environment, so they're germ-free or whatever. So they all have some kind of autoimmune deficiencies. So can I just jump to a conclusion that more diversity in our microbes in our body means that you are a healthier person? Or stand for that?
So diversity has been implicated in a number of diseases. I always like to be careful because I think it's function, right? But by and large, most experts would say diversity is one of the key factors in your microbiome and the most important one. Right? We see this in a number of diseases. I've done a number of studies as well where we see healthy has a higher diversity than unhealthy, right? But again, I think that it has to do with the function, and I think that to some degree that the diversity just gives you a better chance of having what you need, right? I think you can still have low diversity and still have what you need, it's just that your probability is lower.
Is there a geographical difference in the microbiome of people?
Yes, there is a huge biogeographical difference. So it's hard to tell how much of it has to do with diet and how much of it has to do with geography. We know certainly different water sources are very different. We know that if you travel from country to country, it's very different. But remember, those also involve dietary components as well. So it's hard to tell, and genetic components usually. So we're still trying to pull these apart from each other.
Where's the biology that this thing comes from? Is that from our mother or from the environment? If that's the case, how do we balance that? For example, as a person, can I get it tested and say, okay, I'm healthy as is? Not yet. When I'm not healthy, how do I tune it back by a certain diet?
So we actually don't know the answer to that yet. And I actually think, you know, yeah, I guess. No, but you actually can go to America and pay $99 and they will profile your microbiome for you. They will also take the data into future studies. And it's a very interesting thing. We don't know how to interpret that information yet, but I will say certain things about transit from your mother. So we have traced babies through their youth, and children who are born of a C-section have a higher chance of hitting an autoimmune disease than children born through vaginal delivery. And children born through vaginal delivery have a very different microbiome in their early days and a different microbiome development over time. And it's gotten so that a lot of clinics have now, if you are doing a C-section, they will take a vaginal swab and put it in the baby's mouth to try to ensure that you hit off on the right microbiome in the beginning.
May I ask a question? Yeah. How many lawyers in this room? Raise your hand. No, in that case, I can ask. Actually, in this way, I know you take antibiotic, you will kill certain bacteria, then link to certain cancer. If you type that rate, it's a gold mine for the lawyer. They can sue the pharmaceutical company. Yeah, for millions of dollars, because that's your cost. Your drugs cost that. You see the advertise, you heard that drugs, good drug, antibiotics is a good drug. Too much is good drugs, is bad drugs. You can call the following number, we can get you a financial award. Right? Yes.
Well, that is actually a very interesting point, and so that is one of the challenges in our study. I won't say whether it's right or wrong, but we have had to embark on our own sometimes because companies are reluctant to necessarily try a probiotic trial, and so you know, in the stance with many companies is with the probiotics is that if you go to a store, you'll see probiotics everywhere. We actually grabbed them off the shelf and tested them. For the most part, they're actually extremely low dose, And so we've learned that companies have this interesting strategy, which is they'll sell you a probiotic, but they'll give it to you in such low dose. So therefore, they can't get in any trouble.
Okay, that's